What we're going to do today is we're going to finish that story that we didn't quite yesterday, and that's the story of slow axoplasmic transport. So I'll recapitulate that story of the rabbit retina, re-emphasize the concept there of the slow axoplasmic transport, and briefly tell you what we know of slow axoplasmic transport right now. Although that field of slow axoplasmic transport still is a bit murky. We don't really quite know exactly what controls it and how it's regulated, but I'll give you some ideas and we'll move on from there. But mostly what I want to spend our time on today is to talk about glia in the central and peripheral nervous systems. Everything up to now has emphasized the idea of neurons, and we talked specifically about the neuron doctrine and made a big deal about how the neuron is the central cell of the nervous system. Well, we're going to modify that a little bit because glia are also really important. Glia have been known since the time of the neurons. The first sections through the nervous system showed glia. And they knew even then that the number of glial cells was far greater than the number of neurons. But even in the early days, even in the late 1800s, they sort of disrespected the glia. And in fact, glia in Latin means glue. And the idea was that the whole nervous system was simply held together by these cells. Well, they've gained some more respect over the years, but not a whole lot. We now recognize what they're doing is that they're maintaining the environment for electrical signaling for neurons. The neurons don't do much for themselves. They basically are highly specialized cells that signal electrically, and we'll spend a lot of time talking about that. But for them to operate as electrical signaling devices really requires the right kind of environment, and that's what these astrocytes do. And we'll walk through some of the regulatory systems in the astrocytes and the oligodendrocytes, which are the two major kinds of central nervous system glial cells. But the long and the short of it, as far as we can tell right now, is that the glial cells are playing an important role in maintaining the environment, particularly maintaining the environment for the extracellular potassium concentration and for regulating the concentration of neurotransmitters in the very narrow gaps between the signaling cell and the receiving cell, between the presynaptic cell and the postsynaptic cell. Now, as I say that, I'm sure in the next 5 or 10 years, and people are beginning to see some of this now, we're going to see signaling within the glia themselves. They do have ion channels, and they do some signaling, and people have observed calcium waves moving through these glia, and many of them appear to be connected by gap junctions. And so there may be a whole separate signaling system among these glia that we're just now beginning to understand. But for our purposes here, and for the way we normally think about glia, we imagine these glia as playing mostly a regulatory role. Now, that's not to be dismissed. I suspect as time goes on, we're going to find many of the so-called neurological diseases, which we have imparted to neurons, in fact, being the result of dysfunction in the glial cells. I think, in fact, we know and have known for years that multiple sclerosis, and we'll talk a little bit about this, multiple sclerosis is a dysfunction in the oligodendrocytes. So, and I think we're going to find many more neurodegenerative diseases that turn out to be difficulties with the glial cells and not with the neurons themselves. So we'll talk through our way through the astrocytes as controlling the environment, particularly the potassium and neurotransmitter concentrations, and then we'll talk about the oligodendrocytes, and the role that we're going to impart to the oligodendrocytes is primarily this role of myelination, where the oligodendrocyte will wrap its plasma membranes consecutively round and round and round an axon to provide an insulating layer around those conductal regions. And so we'll talk a little bit about how that's done and why that's important. There's a third type of glia beyond the astrocytes and the oligodendrocytes called the microglia. But this, in fact, turns out to be a misnomer. The microglia are not really of the same embryological origin. They don't come from ectoderm. They're not like the glia. They're not like the neurons. These are cells of the immune system. And when we talk about the immune system, we'll find that the central nervous system is what they call immune privileged. There are tight junctions between the capillaries in the brain and spinal cord which don't allow ready access to the immune system into the central nervous system. And so what you find then is that our only line of defense for the immune system in the central nervous system are these microglia, which are in fact macrophages. And in fact, there's a problem with this, because if you get some sort of an infection, viral or bacterial, into the parenchyma, into the tissue of the brain, then the only line of defense against that invasion are these macrophages. And macrophages are not very delicate about how they deal with infections. They tend to emit all manner of oxidative molecules, and those oxidative molecules now can play havoc with the neurons in the glia. So we pay a big price for not having access to the central nervous system by the immune system. There are probably reasons for that. We don't want to have autoimmune diseases attacking the nervous system, but on the other side of that coin is these macrophages tend to be damaging when they're activated, but it's the only line of immune response we have in the central nervous system. We'll leave the microglia mostly behind, and then we'll talk about glia in the peripheral nervous system. And initially, the way I'll draw this is we'll try to contrast those glia which we find in the central nervous system, which we find in the peripheral nervous system. So again, when you see central nervous system glia, think oligodendrocytes and astrocytes. When you think peripheral nervous system glia, think these cells called Schwann cells. Now, the Schwann cell is named after a 19th century histologist. I'll leave this name because I find it easier to say than neurolemocyte. But officially, these cells have been renamed as neurolemocytes to avoid this eponymous naming system. But let's call them Schwann cells. The Schwann cells will myelinate in the peripheral nervous system. So in the peripheral nervous system, they play the role of the oligodendrocyte in the central nervous system. But they have a different embryological origin. These Schwann cells in the periphery then will not only myelinate neurons, the axons in the peripheral nervous system, but they'll also, in some places around the ganglia, they won't myelinate, but they'll actually act almost like an astrocyte. So in the peripheral nervous system, the Schwann cells play both the role of an astrocyte and the role of the oligodendrocyte. And then one of the points I want to make here is that there is a big difference in how oligodendrocytes and Schwann cells operate when neurons are trying to regenerate. Schwann cells are supportive of neuronal generation. That's why you can damage a neuron in the periphery and it will reinnervate its old targets. But if you damage neurons in the central nervous system, they're terrible at reinnervating their old targets. And that problem is largely because there are proteins on the oligodendrocytes which inhibit growth. And this has turned out to be a big problem in trying to solve the difficulties of spinal cord trauma and spinal cord damage and getting the spinal cord to rewire to its old sites. The oligodendrocytes seem to be in the way, and there's probably a reason for that, and we'll talk a little bit about it. But let's start here with where we were yesterday, a short segment here where we were emphasizing the retina, where this was the optic nerve, and this is the retina. And we said that the importance of the preparation was that in the retina there were these neuronal cell bodies of ganglion cells. To be correct, we should call these retinal ganglion cells. And the beauty of the system is those neuronal cell bodies send processes out into the optic nerve. And the processes then had output regions. And I think I misnamed this yesterday. Have output regions in a region of the brain called the thalamus. It's actually in a subregion of the thalamus called the lateral geniculate, but let's not get into that. The major point that I wanted you to recognize is that if these are the cell bodies, then all protein synthesis occurs here. So what they could do then is they could inject radioactive amino acids. So injected radioactive amino acids into the vitreous. Those radioactive amino acids had access to the ganglion cell neuronal cell bodies. That meant that these ganglion cell neuronal cell bodies would make proteins. But those proteins, because they were synthesized from radioactive amino acids, those proteins would be radioactive, so they now had a tag. And what they could do then is that they could plot time versus radioactivity. In fact, let me change that from time and make this a little clearer. So they could plot distance versus radioactivity. And what they could do then is they could inject the radioactive amino acids, they could wait a little bit, they could wait variable amounts of time, kill the animal, and then dissect out pieces of the optic nerve and ask, where's the radioactivity? And since they knew how long it was from the time they injected the radioactivity to the time they killed the animal, they could then estimate how fast the molecules are moving by noticing the distance that they had moved in a particular period of time. So simply put, when they did that, they found two major peaks of radioactivity. One peak they called the fast rate. And this is the one that came up in the neighborhood of about 200 to 400 millimeters per day. And then when they began to explore what kind of proteins these were always the vesicular proteins. Or at least predominantly the vesicular proteins. And then when they looked at this peak that was moving at a much slower rate, and by killing the animals at different times and watching the fast peak move away from the slow peak, they then got a number here of about 0.5 to 2 millimeters per day. Now the numbers aren't important here. What really is important is the disparity. The idea that this fast rate is running upwards of 100 or more times as fast as the slow rate. We know that the fast rate is dependent upon kinesin and microtubules and vesicles. The slow rate now, because we knew what proteins were here, the slow rate tends to be almost entirely cytoplasmic proteins. Particularly, these are proteins of the cytoskeleton. For a long time, it wasn't at all clear how these were moving. Why do we have such discrepancy in the way we move the vesicular proteins from the cytoplasmic proteins? And in some cases, they thought it was simply there was something in the neuronal cell body, like pushing toothpaste out of a tube of toothpaste was pushing these proteins down the axon. That turned out not to be true. And with some advances in microscopy, they then could sort out the proteins moving in these two components. And the surprising finding was that this slow rate moved intermittently. So the molecules in the slow component moved now and again. But when they were moving at the same rates as the fast component. And it now appears that when these move, when these proteins are moving, they're moving by kinesin on microtubules, and that they're moving at the same rates as the fast ones. We still don't know how this happens. One vision of it is that we cut out a section of the cytoplasm, sort of a fabric of the cytoplasm, load that like a raft onto the microtubules, and move that in one unit. But when it's moving fast. But then it stops and sits there for some time. The analogy that I like, one that a few years ago a TA came up with, is that this difference between fast and slow transport is a little bit like hitchhiking. It's as though you go out to I-25 and you decided you wanted to go to Albuquerque. And so you stick up your thumb and you wait for someone to pick you up. So you stick up your thumb, someone stops, they pick you up, and the conversation is, where are you going? And they say, well, I'm going to Berthoud. And so you get in the car and you go to Berthoud. But while you're in the car going to Berthoud, you're moving at the same rate as all the other traffic. But then you have to stop and get off in Berthoud. So you get off in Berthoud and hang out there for a little while, stick your thumb up, and someone comes along, and then maybe they say, well, I'm going to Denver. And so you get in the car and you go to Denver. But while you're in the car, you're moving the same rate as the rest of the traffic. But you have to stop in Denver and hang out for a while. And this then is iterative. Each time you go a distance, you stop, get out of the car, and wait. So in aggregate, you may only be moving a few miles an hour because you're only moving now and again. But when you're in the car, you're moving with the rest of the traffic at fast axoplasmic transport rates. So it's the same idea. And how this is controlled and how we cut out pieces of the cytoplasm, get them to move together in one unit, and then stop is not at all clear. But nonetheless, we do know there are two components. We know that this is vesicular protein. We know that this is cytoplasmic protein. We know that when these are moving, they move at fast rates, but they only move intermittently. So any questions on slow and fast axoplasmic transport? Well, if not, let's head off to glia. So glia. The term glia comes from the Latin. It means glue. And it's because the early histologists thought that all these cells were doing is sort of providing a structural element to hold the neurons together. We now know that the glia come in two great varieties. There are the central nervous system glia and the peripheral nervous system glia. The central nervous system glia come in two major types. These are the astrocytes and the oligodendrocytes. In the peripheral nervous system, we have only Schwann cells, and the Schwann cells, which are also known as the neurolemmacytes. And in the peripheral nervous system is the Schwann cells that are responsible for forming the myelin sheaths around axons or conductor regions. That role in the central nervous system is played by the oligodendrocytes. So it's the oligodendrocytes in the CNS that ensheathe axons and conductor regions. With myelin. In other words, both the Schwann cells and the oligodendrocytes are said to be myelinating cells. The astrocytes play a slightly different role in the central nervous system. In the central nervous system, they regulate the extracellular environment. There doesn't appear, for the most part, to be quite the same kind of corollary for Schwann cells and astrocytes in the peripheral nervous system. Some of that probably has to do with the fact that in the peripheral nervous system, you have large diffusion pathways. You have relatively small amounts of neuron out in the peripheral nervous system compared to the rest of the tissue, and things can diffuse in and out. So you have large volumes in which the neurons exist. That's not true in the central nervous system. In the central nervous system, in order to pack as many cells as possible inside the brain case, inside the spinal column, once we get that many neurons in there, there's very, very little extracellular space. And until you've really looked at sections cut through central nervous system tissue, you don't recognize what a small proportion of the volume in the central nervous system is occupied by the space outside the cells. So one of the important ideas in the central nervous system is in the central nervous system, the volume of extracellular space is limited. Well, because the volume is limited, this means that we can get vast swings in the concentration of ions. And we'll see here a little later that it's the concentration of ions inside and outside the cells, inside and outside the neurons, that's so important for signaling. And so what we have here then is we have, with the astrocytes, a means of controlling or regulating the extracellular environment. Now, when I say that, what I mean primarily is that the astrocytes regulate extracellular potassium concentrations. The potassium concentration on the outside of cells needs to be maintained in the neighborhood of about 4 to 5 millimolar. If the potassium concentration on the outside of cells gets much above this, if it starts creeping into the 8 or 10 millimolar range, then it has drastic effects on the signaling capacities of neurons. We'll see why that is a little later. It has to do with the fact that the membrane potential is due largely to a potassium battery. If you start putting too much potassium outside, you foul the potassium battery. And so what we need to do here is maintain the concentration of potassium within these very narrow ranges. And that's the job of the astrocytes. The other thing that the astrocyte does, and I'll put this over here, is that the astrocyte regulates the concentration of neurotransmitter at synapses. Now I should specify the concentration, the extracellular concentration of neurotransmitters. And we can now come back to what we talked about yesterday and introduce just briefly here the idea of synapses. So if this is a neuron, and this is the output region, and in black, right next door, here is the input region. So we could sort of make it one of the spiky pineapple things if you like. But this is meant to be input. The path here for the neurotransmitter is the neurotransmitter is packaged in vesicles. And when the vesicle membrane fuses with the plasma membrane, those neurotransmitter molecules are released out into this narrow gap between this cell and this cell, between the releasing cell and the receiving cell, between the axon terminal and the input region of the neighboring cell. So now we have neurotransmitter out here. Again, these volumes are precious small. This distance is no more than about 50 nanometers, not much bigger than the width of the vesicle. So that means if we have a fairly small number of vesicle fusions in here, we can shift the concentration of neurotransmitter in this small volume by a whole lot. So the player here is that surrounding the synapse, try to do this so we can get a sense this is one cell. This cell is meant to represent, this is a branch of an astrocyte. So this synaptic site, the synaptic site between the presynaptic cell and the postsynaptic cell, the releasing cell and the receiving cell, that entire volume is now wrapped by this astrocyte. Then there are transmembrane proteins on the astrocyte here, which are neurotransmitter transport proteins. So that the neurotransmitter, when it gets out here in the narrow cleft, can be then taken up and put back into the astrocytes. So basically, you can think about the astrocytes as playing this role of clearing the synapses of excess neurotransmitter. And they do that by having high affinity binding sites for the neurotransmitter and then removing the transmitter from the outside world into the astrocyte. Now, for the sake of completion here, the most common of the neurotransmitters in the central nervous system is glutamate. The glutamate is moved into the astrocyte, and the astrocyte then, the glutamate is converted to glutamine. Glutamine is not effective in opening the receptors. Where glutamate would have opened these receptors here, glutamine doesn't do that. So now the astrocyte can release glutamine. It releases the glutamine, and the glutamine is taken back up into the presynaptic cell, converted back to glutamate, and in that way recycled. So this is a recycling system. The neurotransmitter that's active is released from this cell, binds to receptors, and causes a change in electrical signaling in the postsynaptic cell. But if there's too much of it out here, it's going to overstimulate the cell. So what we do then is we have molecules that will sweep up this neurotransmitter, put it into the cell that's controlling the neurotransmitter concentration here, the astrocyte, and then hand that or convert that molecule into an inactive neurotransmitter and hand it back to the neuron as an inactive neurotransmitter, which it then reconverts into an active transmitter. So you have this cycle. So any questions on that? Hold on to the idea that the astrocytes are really important in regulating the extracellular concentration of potassium and neurotransmitters. That's fundamentally their role. Yes?
So that astrocyte is literally just a ring around the basically gland.
So if you think about the astrocyte around as a ring around the two characters forming the synapse, that'll do fine. In fact, it's not quite that simple because these astrocytes will have lots and lots of branches. They're named astrocytes because people thought they looked like stars because they had so many branches to them. And so the star, the branches of the star will go out and wrap around the synaptic site. It won't be one astrocyte to one synaptic cleft. Okay?
Then how many synapses does it actually encase then as a star? Like 5, 10?
I don't know how many it actually encases. It's more than one, and I'll bet it could be around 100. But I'm picking those numbers out of the air. So I don't think it's 1,000. So an order of magnitude, I'm guessing it's between 10 and 100. Pick a number. Right? Anything else? So well, hold on to the idea that that's what the astrocytes do. They're environmental regulators. And as time goes on, if you revisit any of this material, remember that regulating the environment is really important in the central nervous system. And if there are pathologies that are going to affect the astrocytes, they're likely to affect the local environment. And by affecting the local environment, then the neurons won't function as well anymore. And so I think we're going to see as time goes on, more and more central nervous system diseases, which we now ascribe to problems in neurons, being ascribed to glial diseases, particularly among the astrocytes. What I want to do on this panel is talk about the role of the oligodendrocyte. And we'll spell this out to say the oligodendrocyte oligodendrocyte forms wraps around axons, which you can also read as conductor regions. So we'll start here by imagining an oligodendrocyte in development forming the myelin sheath. And I'll do this by simply taking a circle and labeling this circle as the plasma membrane of the axon or conductor region. Then up here in blue, I'm going to put a cell body of a cell I'm going to label here the oligodendrocyte. So in development, the oligodendrocyte will send out a tongue of cytoplasm which is bounded by the plasma membrane. I wish I'd started farther left. This oligodendrocyte sends this process out and it contacts the plasma membrane or the axon. And there are almost certainly sticky proteins out here that allow the oligodendrocyte plasma membrane to stick to the plasma membrane of the axon. But once the oligodendrocyte membrane sticks to the axonal membrane here, it then continues to grow. So it will remain attached, but it continues to grow so that it looks a little bit like this. So again, this cell body of the oligodendrocyte is up here. The process has been sent out, and it's grown around the axon. So if you can picture this, what I'm trying to show is that the plasma membranes of this oligodendrocyte have effectively circumnavigated the axon. They've grown all the way around the axon. And when they grow around the axon and they come back to this point, they're going to meet the neck of the oligodendrocyte here. Now, when that happens, I'll try to see if I can squeeze one more in here. When that happens, this leading edge will sneak under the previous circumnavigation, so that we'll end up with this leading edge Sorry for being so slow on this, but I always foul this up. So this is the cell body. And if you follow this out, and I've done this right, we should be able to follow the cytoplasm all the way around here. Back to the leading edge. So again, the leading section of this oligodendrocyte contacts the neuronal plasma membrane, sticks to it. But doesn't stop there, it continues to grow. It continues to extend. But the way it grows is to remain stuck to the plasma membrane of the axon. It grows around, and now at this point, it runs into its start point. But now what it does is it forces this previous plasma membrane of the oligodendrocyte away from the axon and sneaks underneath. So we can extend this a little bit to see how it's going to extend underneath. It sort of squeezes its way underneath and then continues to grow. And as it continues to grow now, it wraps around the axon many, many times. And each time it wraps around the axon, it squeezes in under the previous wrap. But what we have here now is a continuous layer of cytoplasm bounded by the plasma membranes. And each time this neuron grows around the axon, it leaves behind those two plasma membranes and squeezes out the cytoplasm in between. So in the end, what you end up with is something that looks a little like this. There's the axon. And then there is layer upon layer. And I won't try to maintain the continuity that I have on the right side. There's layer upon layer of plasma membrane. And this is just the plasma membrane of the oligodendrocyte. But in so doing, this generates a kind of insulating layer. After layer around the axon. These layers are called layers of myelin. But in reality, what the layers of myelin are the plasma membranes. Of oligodendrocytes. So the plasma membrane of the oligodendrocyte now, by over and over again, wrapping and wrapping and wrapping, makes as though you were taking electrical tape and wrapping it around a wire. Over and over and over again, providing an insulating layer. Well, this is fine for thinking about how the oligodendrocyte is organized in this cross-section. But you have to recognize that if this is a neuron and this is an axon, this axon is coming out into the audience at this dimension for hundreds of meters, and behind the board, hundreds of meters. So this gives us one oligodendrocyte wrapping around the axon. But what I want to do now is change the orientation. So where we're looking at this as a cross-section and looking down the axis of the axon, now what I want to do is take this and turn it at right angles and think about what this image is going to look like if we view it at right angles. So we'll go up here to the upper panel and we'll label this panel as the longitudinal view of oligodendrocytes. So rather than showing the circle as we have above, we're going to make two parallel lines. And the two parallel lines, again, will represent the plasma membrane of the axon. So these are meant to be the plasma membrane. These are meant to represent the plasma membrane of the axon. Now, if we were to take this and not think of it as a cylinder, but think of it, we've cut down the cylinder and we're looking inside the cylinder. So what we're looking at here in purple, this is the cytoplasm of the axon. Sometimes this is known as the axoplasm. But now, to be consistent with what we have in the upper board, we see these concentric rings of myelin. These are the plasma membranes of the oligodendrocyte. But now, rather than viewing them in cross-section, we're viewing them in longitudinal section. And what we find is that if we think about the distance along the axon that these oligodendrocytes extend, this distance is in the neighborhood of 1 to 1.5 millimeters. And then at these intervals of 1 to 1.5 millimeters, we see there's a break in the myelination. So every interval of about a millimeter or a millimeter and a half, there's this opening. These openings have a name. The openings are referred to as the nodes. Again, this is an eponymous name, nodes of Ranvier. Ranvier was a French histologist and pathologist in the 19th century, and we still name these structures the nodes of Ranvier. But what the nodes of Ranvier are, what they in fact are, is they are plasma membrane. Plasma membrane of the axon facing the outside world. So everything out here is extracellular. The regions with these blue stripes on them, these regions have a name. These are called the internodal regions. So these internodal regions are characterized by the consecutive rings or concentric rings of myelin that go all the way around the axon. They're of a limited length, a millimeter or so. And then there are these openings called the nodes of Ranvier. Now I'll put it in here, and we'll come back to this in much more detail later. But at these so-called nodes of Ranvier, we have clusters of voltage-gated ion channels. For the most part, there are few voltage-gated channels here in the internodal region. So the internodal regions, this is a bit of an exaggeration, but they mostly lack voltage-gated ion channels. That's simply to say that we're going to cluster the voltage-gated channels here at the nodes of Ranvier where we have naked plasma membrane. And we're going to do this at intervals all the way along the axon. And again, concentric rings. We do this over and over again. And if, for round numbers' sake, we have a meter-long axon and we have millimeter distances between the nodes, we'll have 1,000 of these along a meter-long axon. What we're going to see a little bit later is that the electrical signaling is going to jump from node to node. But all I want you to notice now is we have these specialized cells, the oligodendrocytes, that form these phospholipid bilayer wrappings around the axon to provide an insulating layer. We'll come back to this a little later because we're going to see that this is going to be really, really important in increasing the rate of propagation of axons, or of action potentials. Action potential propagation in these kinds of axons could be at the rate of about 110 meters per second. Again, as I alluded to yesterday, recognize that this is the propagation of an electrical signal at over 100 meters per second, whereas the propagation of the the fast and slow exoplasmic transport going on in the cytoplasm is a fifth of a millimeter at its fastest. So there's an enormous difference here, and that's because we're going to be moving ions across the membrane for this story, and we're removing proteins for the other story. So any questions up to here? If not, let me take a couple of minutes and show you just a couple of slides quickly. The first slide is to summarize the exoplasmic transport story, the idea that we have fast and slow exoplasmic transport, and that the fast transport is all carried on vesicles on microtubules. And that's sort of the story here, and I won't belabor that anymore. What I want to show, though, is this sort of picture, a diagram of an astrocyte. And the astrocyte here is showing an extension that comes down to the synapse. Now, they don't show this extension as wrapping all the way around the synapse, but in fact, it would wrap all the way around. It would girdle the synapse and provide a site where we could regulate the neurotransmitter. All along this region, out in the extracellular space, there will be pumps here to maintain the sodium and potassium gradients. So that's really, really important for maintaining the gradient with these cells. And then finally, and we didn't even discuss this, that many of these astrocytes will have processes that go from the neuron all the way out to a capillary. And it appears that they'll now move molecules through the cytoplasm and out to the capillary. So they're regulating the extracellular concentrations by moving excess molecules out to the blood and getting rid of them in the blood. Then here, this is a picture I like. It looks like Starship Enterprise, and this is meant to be this is meant to be the cell body of the oligodendrocyte, and it shows how these jelly roll structures are being sent out and wrapping around the axons. And then finally, let me show you this image. This is a Schwann cell, which we'll just have a couple minutes to talk about. But what I want to show you is this is an animation, and it shows the way, hopefully soon, this is the wrapping and wrapping. So each time it wraps around the axon, it leaves behind the two layers of its phospholipid bilayer, and those phospholipid bilayers now become the lipid-rich myelin sheaths. It provide the insulating layers around the axon. And this is the way these things grow. They just wrap and wrap around. Yes?
So is it that mechanism that's, I guess, deteriorating in people with MS?
Oh, okay. So this is the MS question. I may as well address this now. We don't have much time. All right. No. This is something I wanted to bring up. And it's a great example of how diseases which express themselves as neuronal diseases are in fact diseases of oligodendrocytes. Multiple sclerosis is a disease where people start having difficulty controlling muscle and other activities. And what the disease is it's an autoimmune disease where the immune system, immune cells, recognize proteins on oligodendrocytes as foreign.